The image on the screen shows the memorial of a late Prime Minister of India, Rajiv Gandhi. You might also know that he was assassinated by a suicide bomber. But do you know how the attacker was identified even when the bomb exploded his body into small broken pieces? Likewise, in the famous tandoor case in which the chopped body of the wife was burnt in a tandoor, how did the forensic department identify the burnt remains of the victim? The answer lies in the modern technology based on DNA. You know that the DNA is a genetic material present in the cells of living organisms, including humans. But if the DNA is present in all organisms, how can it help to identify a particular individual even from the burnt remains? The answer is DNA fingerprinting. Let's discuss the technique of DNA fingerprinting in detail to find out the answers. We already know that the human genome is composed of 3 into 10 raised to 9 base pairs. But do you know that the DNA sequences may be present once or repeated in the genome? The DNA sequences that are present only once throughout the genome are called unique sequences, which makes up around 41% of the human genome. The rest 59% of the DNA sequences are repeated more than once and make the repetitive DNA. You might ask a question here. Why so? Let's take an example. You know that the ribosomes are composed of rRNAs and proteins. To synthesize proteins, cells require ribosomes. So, the genes that code for different types of rRNA are present in multiple copies in the human genome. On the other hand, the gene that codes for lysozyme present in human tears is present in a single copy. You might be thinking, what is the relevance of repetitive DNA here, right? To understand, let's first discuss the repetitive DNA in detail. Based on the number of repeats, the repetitive DNA is grouped into many categories. The satellite DNA is one such category and constitute the DNA sequences explored for DNA fingerprinting. Do you know why it is called satellite DNA? Density gradient centrifugation is a technique that is used to analyze the density of DNA. When the human DNA is analyzed by the density gradient centrifugation, the majority of DNA is present as a single band or peak and is called the main band DNA. However, some DNA sequences make additional bands or peaks as they differ from the main band's DNA in density. These DNA sequences are composed of satellite DNA. You might ask a question here. Why is the density of the main band and the satellite DNA different? The density of DNA depends on the percentage of GC and AT base pairs, that is guanosine, cytosine and adenosine and thymine base pairs. So, different percentages of these base pairs impart different densities. Satellite DNA is repetitive DNA. Minisatellites and microsatellites are the two different types of satellite DNA. They differ from each other in the number of repeats, the percentage of AT and GC base pairs, and their length. The mini-satellites do not include any protein coding genes and are mostly composed of non-coding sequences present within or between the protein coding genes. It includes variable number tandem repeats, that is VNTRs. The number of copies of a specific VNTR present at the specific sequence varies from one individual to the other. So, variation in the size or length of these VNTRs was used for DNA fingerprinting. I have a doubt here. So, what exactly is DNA fingerprinting? And what about the microsatellites? Let's discuss the microsatellites first. Microsatellites are the repetitive DNA sequences that are also dispersed throughout the genome, and the individuals differ from each other in the number of repeats of a particular microsatellite sequence located at the specific site in the genome. These are smaller than mini-satellites and are now preferably used in DNA fingerprinting over the earlier used microsatellites. Before discussing the technique of DNA fingerprinting, so let's discuss some related concepts. Humans are sexually reproducing organisms, and the repeated division in zygotes form the multicellular body of an organism. So, all the cells present in the body of an individual have identical DNA sequences. This means that all the body cells of an individual would have the same repeats and number of microsatellites and mini-satellites. The variation in the DNA sequence is called the DNA polymorphism. Here, the microsatellites and the mini-satellites exhibit polymorphism for their length or size among individuals. The polymorphism in these DNA sequence is transmitted from parents to their children. This means that if a parent has a particular microsatellite of a specific length at a site in the genome, the child would also have the same microsatellite of the same length. I have a doubt here. How does this polymorphism in DNA arise? And the answer is mutation. Recall that mutation is a random change in the DNA sequence of organisms and give rise to new alleles or new alternative forms of the gene. Now, if a particular locus on a chromosome has more than one allele, it is called DNA polymorphism. The frequency of this variation in the allele in the human population is more than 0.01. The variation is transmitted from parents to children. This is possible only when the new allele is not harmful and allows the organism to survive and reproduce successfully. Let me ask you a question here. Now that you know about the protein coding DNA sequences and the non-coding sequences, should the DNA polymorphism be more prevalent in coding or non-coding sequences? And why? Let's check out. The non-coding DNA sequences such as microsatellites and mini-satellites do not code for proteins, and therefore mutation in these sequences are least likely to affect the survival and the reproduction of the individuals. And this is why these non-coding sequences exhibit a high degree of DNA polymorphism. I have again doubt here. How is polymorphism in these satellite DNA sequences used in DNA fingerprinting? And again, the same question, what is DNA fingerprinting? You know that the fingerprints of different individuals are unique, right? DNA fingerprinting is the technique of analysis of the unique DNA sequences present in the genome of an individual. It was developed by Sir Alec Jeffreys in 1984. You might be thinking about how it works, right? And what are the steps involved in DNA fingerprinting? The first step in DNA fingerprinting is the isolation of DNA to analyze it. The isolated DNA is then broken down into smaller fragments using restriction endonucleases. Restriction endonucleases, and what are they? They are a special type of DNA digesting enzymes that break the DNA at a specific site. Each restriction endonuclease identifies a particular DNA sequence and cleaves the DNA within that sequence. So, the digestion of DNA with a particular restriction endonuclease would break it in all the regions where the sequence identified by that enzyme is present. The DNA fragments produced by the digestion with restriction endonuclease are then separated from each other using the technique of gel electrophoresis. You might ask a question here. What is gel electrophoresis? Recall that DNA is a negatively charged molecule due to the phosphate groups. Gel electrophoresis uses a gel to which an electric field is applied so that the negatively charged DNA fragments move towards the positive side of the gel. The speed of the movement through the gel depends on its size. The larger DNA fragments move more slowly than the smaller ones. This is how the DNA fragments are separated from each other. The separated DNA fragments are then transferred to the DNA binding filter or a membrane, and these membranes are composed of nitrocellulose or a nylon and binds to the DNA fragments so that they are positioned in the same location as they were in the gel. And this process is called southern blotting. Here, the DNA fragments on the gel are transferred onto the yellow colored filter. The membrane with the DNA fragments is then incubated with a radio-labeled VNTR probe. So, what is a probe? A probe is any single-stranded DNA or RNA molecule that identifies the DNA or RNA with a complementary sequence. Here, the VNTR with a small sequence is used. Since the number of copies of a VNTR present on different chromosomes of an individual varies, if the membrane is visualized using an autoradiography after the hybridization of DNA fragments on the membrane with a radio-labeled VNTR probe, DNA appears as a series of bands of different sizes on the gel. In autoradiography, the membrane is washed to remove the excess probe and then exposed to X-ray film. Due to the variation in the length of a particular VNTR at any location among individuals, the DNA of different individuals produces different patterns of these bands. And the unique pattern of DNA bands of an individual is called its DNA fingerprint. So, what are the applications of DNA fingerprinting? In other words, where is the technique used? Recall that DNA from any cell of an individual has the same polymorphism. So, DNA fingerprinting of DNA taken from any cell of an individual would produce the same unique pattern of bands. On the other hand, the pattern of bands would be dissimilar if the DNA is taken from a different individual. This is how DNA fingerprinting helps in the forensic identification of victims and criminals using the DNA from hair, saliva, sperm, blood stains or burns remain of the body. Here the DNA fingerprint created by the DNA sample obtained from the crime scene is compared with the DNA fingerprint of 3 suspects. The pattern of bands of suspect 2 is the same as that of the crime scene DNA sample. Did you notice the DNA from hair or the blood stains are taken? Can a piece of hair or even a long strand of hair obtain enough to DNA for creating fingerprints? The answer is yes. It is possible due to one more modern technique called polymerase chain reaction or PCR. PCR produces multiple copies of a small sample of DNA. So, the DNA samples obtained from the hair or blood stains can be amplified using PCR or the polymerase chain reaction, and the resultant multiple copies of the DNA are then used for DNA fingerprinting. Let me ask you a question here. Does DNA fingerprinting distinguish the monozygotic twins who are born from the same zygote and therefore share the identical DNA sequences? The answer is no. The DNA from monozygotic twins would produce the same pattern of bands and therefore the technique cannot be used for the identification of monozygotic twins. I have another question here. Is there any other application for this technique? Yes, there is. As already mentioned, polymorphism is inheritable from parents to children. Analysis of DNA fingerprints of suspected parents and a child also solves the dispute about parentage. Here, the child shares pink-colored bands with the mother and blue-colored bands with the dad. DNA fingerprinting is also used to study the population and genetic diversity of endangered species and other life forms. Now that you have learned about the DNA and applications of DNA technology, can you imagine how different life forms that share common features such as the presence of DNA as genetic material have evolved on the earth? Did the earth have the same species of plants and animals millions of years ago, or there was a change in the flora or fauna of the earth? In other words, how did life forms evolve on the earth? Let us find out answers to these questions in upcoming videos. Now, let us summarize what we have learned.
Manorama Horizon. Learning made simple.